Bellen, how did she beat me?
Well, have you ever wondered what takes place during a muscle contraction?
Well, I want to now.
Well, let's take a look. The muscles are made up of bundles of muscle fibers, myofibers. These bundles form fascicles. Now let's look deeper into one of the myofibers, one of the muscle cells. One muscle cell is made up of a bundle of myofibrils that are surrounded with a membrane called the sarcolemma, surrounded here in the clear plastic. Now let's look under the sarcolemma so that we can see what's inside. We can then see the sarcoplasmic reticulum that surrounds each myofibril, shown here in purple, which stores calcium with the molecule calcium, seen here with the white beads. This holds on to calcium. In addition, there are also mitochondria, seen here in red, which is essential in ATP production. Also, there are T-tubules, which is an invagination of the sarcolemma membrane that covers the myofiber. So now let's see what's inside a myofibril. The basic unit of a myofibril is a sarcomere. The Z-discs are protein that make up the 3D space and separate each sarcomere. The A-band is the area that is the darkest, containing the thick filaments. The I-band is the light area that contains the thin filaments. And the H-band is located in only the area that contains the A-band, or the thick filaments, and has no thin filaments. This makes up the basic fundamentals of every muscle cell. So now that we have seen the basic structure of a muscle cell, let's look and see what happens during an action potential, in which the muscle is contracted. An action potential is created by depolarization of the cell membrane, in which the ions of sodium move across the motor end plate, causing the inside of the synaptic cleft to become more positive and the outside to become more negative. In the action potential, calcium ions, seen here in green, flood in through the channels of the axon. The calcium pushes the synaptical vesicles down the axon, and then the synaptic vesicles fuse with the axon membrane, spilling out acetylcholine, the red molecule seen here on this model. The acetylcholine pours into the synaptic cleft, in between the axon and the motor end plate, seen here in yellow. The acetylcholine then attaches to acetylcholine receptors, seen here in orange. The receptor opens and allows sodium, the pink molecule, to flow through the motor end plate into the muscle cell, and then allows for potassium, the yellow bead, to move past the motor end plate into the synaptic cleft. Acetylcholinesterase, the yellow molecule seen here, which is also found in the synaptic cleft, is an enzyme that breaks down acetylcholine into two molecules, stopping the depolarization of the cell membrane. The sodium in pink drives the action potential down through the T-tubules, which triggers the release of calcium in the sarcoplasmic reticulum. The myofibril is made out of thin and thick filaments, which drives each muscle contraction. The thin filament is made up of three things called the tropomyosin complex. Tropin, which is seen as the silver clamps, and the blue tubing represents tropomyosin. The white bulbs represent actin protein. Calcium ions that come out of the sarcoplasmic reticulum then attach to troponin. Here we have the tropomyosin complex. The calcium triggers the troponin to be pulled off of the tropomyosin in blue. Pulling off the tropomyosin results in exposing the active site, seen here as a clear dot, which is found on the actin protein. The thick filament is made up of myosin, which consists of a head and a tail, seen here in silver and blue. The myosin head has a contracted and relaxed state. The excited state is created when a molecule, ATP, seen here in blue, attaches to the tropomyosin head. The ATP distributes one of its phosphates onto the myosin head. that energizes the myosin head and converts ATP to ADP, the light molecule seen here. By exposing the active site on the actin protein, the myosin has an uncontrollable urge to bind to the active site. Another ATP molecule, seen here in dark blue, attaches to the myosin head that detaches the myosin head away from the actin protein. This process is replicated over a series of times that leads to a full contraction.